Let's take a closer look at what transposable elements are and how they function, the problems that they cause, and the biblical evidence that sin is ultimately a genetic problem. Transposable elements are sequences of DNA that can move around in the genome. They are classified into two major groups, Class I and Class II transposons. Class I retrotransposons copy and paste themselves in the genome. They do this by using the cell's existing machinery to create RNA and enzymes called reverse transcriptases. The cell transcribes the retrotransposon's code into RNA. The reverse transcriptase then synthesizes a new DNA copy from the RNA and then inserts the DNA copy at a new location in the genome. A little side note on reverse transcriptase. The cell's normal usage of genetic code is that DNA is transcribed into RNA, which gets translated into proteins, never the other way around. Reverse transcriptase upends this by allowing DNA to be synthesized from RNA, reverse the normal process. It's interesting to note that the code for reverse transcriptase originates from transposable elements. Furthermore, reverse transcription is very error-prone, frequently halting partway through transcription. I would argue that these characteristics indicate inferior design. Anyway, there are two subtypes of Class I retrotransposons called long terminal repeat or LTR transposons and non-long terminal repeat or non-LTR transposons. LTR transposons are so named because they are flanked on either side by long repeating sequences. Retroviruses are viruses that insert themselves into the host's genome and are classified as LTR transposons because they share many of the same characteristics. Retroviruses are basically LTR transposons that encode for a protein envelope allowing movement between cells. HIV is an example of an LTR transposon. The second subtype, called non-LTR transposons, consequently lack terminal repeats and are further subgrouped into lines or long interspersed nucleotide elements and signs or short interspersed nucleotide elements. Lines are 4,000 to 7,000 base pairs long and are autonomous, which means that they encode the enzymatic machinery to move themselves around the genome. Line-1s are the most common lines in mammals. Signs are 150 to 500 base pairs long and are non-autonomous, meaning signs can't move themselves and must rely on the enzymatic machinery from lines already existing in the genome to mobilize. Both lines and signs can be identified by a repetitive tail and the lack of long terminal repeats. In fact, all transposons can be identified by various forms of repetitive sequences. Class II transposons, called DNA transposons, cut and paste themselves in the genome. They do not rely on an RNA intermediary and instead rely on several transposase enzymes which perform the cut and paste operation directly on the DNA itself. Some transposase enzymes are very specific in the sites that DNA is pasted to, while others are not very specific, pasting DNA at random. DNA transposons can be identified by terminal inverted repeats, which are short direct repeats at the target paste site, followed by inverted repeats which help the transposase identify and cut the transposon. DNA transposons make up about 3% of the human genome, but are thought to be non-functional in humans due to losing the ability to transpose. That was just a brief overview in how transposable elements function. The point is that transposable elements are sequences of genetic code which move themselves to other places in the genome, and that they can be identified by repeating sequences. Most scientists would argue that transposable elements provide a vital means for genetic diversity and genetic mutation to facilitate evolution. However, I don't think that this is the case. I believe that all changes caused by transposable elements are degenerative. Here's why. Transposable elements cause significant damage to the genome and are implicated in a vast array of diseases. Genetic diseases, autoimmune diseases, endocrine and metabolic diseases, psychiatric diseases, selfish behavior, cancer such as lung cancer, brain cancer, ovarian cancer, breast cancer, aging, and death all have root causes implicating transposons. Most alarming is the fact that transposons are very active in brain cells. Line-1 retrotransposition is active in neurons and causes somatic mosaicism, which means that normal cells have different genomes from one another due to random line-1 insertions. The rate of line-1 transpositions is disputed, but at 80 to 100 billion neuron cells, even the most conservative transposition rates results in extensive genome variation within an individual. Furthermore, normal neuron activity, thinking thoughts, causes double-strand breaks in the genome of neurons. These double-strand breaks occur at locations in the genome where transposable elements are abundant and allow even more transposon insertions at the breakpoint. These breaks change how brain circuits function and even changes thought processes. Basically, transposable elements can alter our thoughts and behaviors. Here's one short example of how transposons have altered behavior. Research was done investigating the difference between monogamous prairie voles and promiscuous montane voles. Researchers wondered why the prairie voles mated with one partner for life and showed more interest in and care for offspring, while the montane voles didn't show the same pair bonding nor care. It was found that prairie voles have greater binding to vasopressin 1a receptors in the brain, while montane voles have less binding at these receptors. Vasopressin 1a receptors are necessary for societal and emotional behaviors such as forming pair bonds in voles and also in humans. Thus, the higher receptor activity results in monogamous behavior in prairie voles, while lower receptor activity results in promiscuous behavior in montane voles. Researchers further showed that variations in vasopressin receptors correspond to changes in behavior by altering the expression of the receptors in the voles. Previously monogamous prairie voles showed promiscuous behavior when the researchers downregulated vasopressin receptors, while previously promiscuous montane voles showed monogamous and pair bonding behavior when vasopressin receptors were upregulated. The root cause of the variations in the vasopressin receptors in the voles was variations in the length of microsatellite gene regions. Microsatellites are repeating sequences of genetic code caused by transposable elements. Basically, transposable elements cause microsatellites, which cause variations in vasopressin receptors, which cause variations in behavior. Transposable elements affect behavior. What biblical evidence is there to link transposable elements to sin? First, I'm going to propose the following key. When the Bible reads iniquity or unrighteousness, substitute transposable elements. When the Bible reads sin, it means actions caused by transposable elements. Let's look at some examples. Psalms 31:10. For my life is spent with grief and my years with sighing, and my strength fails because of my iniquity, transposable elements, and my bones waste away. Psalms 38 verses 3-4. There is no soundness in my flesh because of your anger, nor any health in my bones because of my sin. For my iniquities, transposable elements, have gone over my head. Like a heavy burden, they are too heavy for me. These two verses are describing sin and iniquity as physical and tangible things which are weighing the psalmist down. Iniquity is a burden which fits the effects of transposable elements. Some others. Psalms 51 verses 1-2. Have mercy upon me, O God, according to your loving kindness. According to the multitude of your tender mercies, blot out my transgressions. Wash me thoroughly from my iniquity, transposable elements, and cleanse me from my sin. In Jeremiah 33:8, I will cleanse them from all their iniquity, transposable elements, by which they have sinned against me, and I will pardon all their iniquities, transposable elements, by which they have sinned and by which they have transgressed against me. These two verses are describing a physical process of cleansing the body of sin and iniquity, as if sin and iniquity are physical things. Finally, the Bible gives many accounts of leprosy. True leprosy was incurable in Bible times. It was contagious, spreading via droplets. It physically deforms the victim. It spreads throughout the whole body. It isolates its victims as outcasts. It attacks the sheath of the nerve cells, gradually causing inflammation, then death of the nerve. There are many parallels between leprosy and sin. Sin is incurable. Sin is contagious. Sin physically deforms its victims. Sin spreads throughout the whole body. Sin isolates its victims as outcasts. Sin causes death. Mycobacterium leprae, the bacteria culprit for leprosy, has been heavily invaded by transposable elements. I'm trying to make the argument that all sin is caused by transposable elements. The blueprint for all life is defined by DNA. The reason we have a head, 2 eyes, a nose, a mouth, a neck, a body, 2 arms, and 2 legs is because these are all coded for in DNA. The reason we have senses to see, smell, taste, and touch is because these are all coded for in DNA. The reason we have brain circuitry to think and act on these senses is because it's coded for in DNA. The reason we have a tendency to lie, cheat, steal, gamble, gossip, covet, and commit adultery is because we have defective brain circuitry, which results from defective DNA. The root cause of sin must be in DNA. Sin is not a legal problem where the sinner has transgressed God's law and needs a lawyer to be justified. Sin is a physical problem where the sinner needs to be healed. God is pleading to let him heal us and fix this transposon problem. The reason God gave us the Ten Commandments is to diagnose us with sin. If we had no transposable elements, we would not break God's commandments. We would keep God's law implicitly. But keeping the Ten Commandments doesn't save us. Removing transposable elements does. Does God punish for breaking his law? No. Selfishness, greed, coveting, pain, disease, cancer, and death are natural consequences of breaking God's law. Finally, with the hypothesis that all sin is caused by transposable elements, we can make a prediction about nature. I predict that all disease is caused by transposable elements.